a person has cancer and there's a tumor. Let's take a sample of the tumor and look at the DNA sequence of the tumor. And then let's take a sample of their healthy cells, could just be a blood cell, look at that DNA and just compare the two. And it's very much an engineering or software things, right? Like I'm going to get two strings of bits and I'm just going to compare them. And most of the time they'll be exactly the same. But occasionally one of them will be different. And you'll immediately go, that's a variant that's somehow involved with the cancer. And so it seemed like a very elegant way to work out what was happening with cancer.
Hey everyone, I'm Costas.
I'm Lester, and welcome to another episode of What's with RWE. And we have an enormously fortunate to have this guest on today, Alex Dickinson. He's actually, I believe, Costas, probably the first non-Canadian that we've had on this podcast. Is that true?
I think that's true. Yeah, but a Commonwealth, character nonetheless.
I'm honored.
Absolutely.
Honored today to be a member of the Commonwealth and maybe on loan into Canada. Thank you.
Absolutely. You're honorary Canadian. We'll grant other Australians that privilege as well. But just give everyone a little background around Alex, and most of our audience, those who are in the genomic space, know Alex very, very well. For those of you who are not familiar with Alex's work, so Alex has been a long time executive in the genomics space and genomics industry. He is now the board of director and advising almost like, I don't know how you do it, like half a dozen companies, biotech companies and tech companies. And probably where many, at least from the genomic side, know Alex very well is he is a LinkedIn provocateur extraordinaire. He has a lot of spicy takes on LinkedIn that are just absolutely amazing. So you want to check some of those out. That would be fantastic as well. So Alex, welcome to the podcast. Thanks a lot for joining us.
Thanks, Lester. Thanks for the lovely introduction. I appreciate it. Very nice. Thank you.
So today is, happy post-election day, and we won't jump into politics because that's going to get everyone super angry. So maybe on that specific thread, though, as a long-time executive in both tech companies and biotech companies, and of course in genomics space, and now advising so many genomics companies and started many companies yourself, did the politics in terms of the elections of who would become president or who would become prime minister or a senator, did that matter to you in terms of how you built strategy or around the company? Or how would you advise, you would say, some of your organizations right now to think about that? Should they just don't care, keep going, or what would you say to those organizations?
Yeah, one of the first texts I got from a friend this morning was actually a friend/journalist, was who's going to buy Illumina now? So I was like, oh, interesting. I think, and I have, because of the Illumina history, followed the antitrust area pretty closely. And I think it's one of the areas that may not change that much. Lena Khan has somehow threaded the needle, and she has supporters both on the left, who obviously appointed her, under the Biden administration, and the right. And so there's some common ground there. And particularly with J.D. Vance coming out of the particular subcategory of Silicon Valley people that he does, I'd say in general they're kind of supportive of that. They're I think, what did Andreessen Horowitz call it? It's like the small things thing, which is they're very sensitive to monopolies. And they feel often that small companies, startups have a real challenge thriving in the face of a Google, Microsoft, Amazon, Meta. And so it's interesting to me that there is that common thread, because once upon a time, it would have been like, well, all the rules are off now. Everyone can just buy everything they want and merge. And it's not clear to me that that's going to be the case. So one thing is, I don't think we know yet, and it'll probably take, I don't know, I guess it'll appear over time. But with this particular class of Republican administration, things seem to change a lot. There's a lot of it's the constant is the change. So we'll have to see. So sorry, very long answer to the question. I think it's too soon to tell. It's not necessarily good news or bad news for startups or large companies. I think we have to see how it plays out.
Completely fair enough. Just want to take a little bit different take than just ask you, oh, what you think about the election itself. But actually, for those of us, the audience, let's say there's billions of people watching right now, which hopefully that'll be the case one day, especially with you on. Actually, maybe we'll be billions, at least hundreds of millions. But for those of you, us that aren't familiar with the genomics space, I mean, that's where how I know yourself, Alex, from the genomics space is like, why should people care about genomics, especially people that everyday life. This is just they're just going about their usual duties of going and going to the office, doing their 9 to 5. Like, why should people care about genomics?
Sure, I think that's a great question. Just kind of set the stage, and I have been thinking about that a lot. I guess it's because it's what we do. I think the part of genomics that got a lot of the early publicity was around the Human Genome Program, and it felt very much like science. And there was a lot of gee whiz in it, and we'll understand DNA and what the sequence is. And there was also a huge amount of optimism that we would cure all disease and everything else around that. And I'm old enough now that I'm starting to get the rhythm of these cycles. And we have a cycle of incredible enthusiasm around new medical technologies. And we're very naive. And then there's a reckoning where we realize that actually everything we thought we knew we didn't know, and that it's some very complex onion that we continue to peel. And so I feel like the heart of genomics certainly started out around the idea that we'll understand the human blueprint. And if you understand the blueprint or the diagram for something, the instructions for building it, kind of the sense that you'll just understand everything, right? We know that you put a double-stranded piece of DNA, a genome, into a cell, and it'll rebuild whatever's in the DNA. I mean, vast oversimplification, right? So it must mean everything is there. And it turned out that that wasn't the case. But we did learn a lot, and we in particular learned a lot, obviously, about cancer, because it's such a huge problem for humanity, and it's a disease of the genome. So I think, harking back to your question, the reason it's important to everyone is that we're only either 0 or 1 degree away from someone that's had a usually traumatic, if not fatal, experience with cancer. And so that becomes very important. And I think the understanding genomics has had a huge impact on not just our understanding of cancer, but us really racing along in terms of precision medicine for cancer, which has had an extraordinary, I think, Jimmy Carter is a great example, right? I can't remember how long ago his cancer diagnosis was. He had an amazing precision cancer treatment, and he is still with us, though I think coming up on 100 years old. Um, and then the other case is, and this was interesting to me as an engineer who kind of stumbled into these things, is understanding that part of it. And there's these two ways to think about it. As you think about a human, you've got your own genome, but then your bloodstream is full of the genomes of all sorts of beasts, viruses and bacteria and everything else. And we saw that during COVID. COVID, by some mechanism or another, emerged in Wuhan, China. And I believe it was first sequenced on an Illumina MiSeq sequencer. And immediately, as soon as you had that, it became perfectly clear that this was a whole new, not whole new, but anyway, it was a very new thing for our immune systems. And that led to the design very quickly of a PCR test, which there were billions of those done. And then over time, of course, led to the ability to assemble those mRNA vaccines. And so the importance of genomes is so profound, both in our own genome, but also the fact that the world is just full of the genomes of every living thing. And every time we scoop up some, um, really any organic sample in general, we're going to get just this incredible mishmash of all those different genomes, and clearly can learn a huge amount about them.
That's interesting. And so you mentioned you just touched on it. Your background is in engineering. So how did you make that leap or make your way to the world of genomics and even I think more recently more on the clinical side? What was that journey like?
Yeah, so I was running a spin out of Caltech, which was an optical silicon chip company, which we founded in 2001. And I was still running it in around 2006. Two profs from Caltech, Axel Scheurer, who was with me at Bell Labs, and also David Baltimore, who got the Nobel Prize for reverse transcriptase.
Mhm.
They were both working on a high performance real time PCR instrument, and they wanted to start a company. And I literally didn't obviously know what PCR was. I never did any biology, certainly no microbiology. But I loved raising venture capital. And I said, oh, well, I'll take a little time out from my day job and I'll help you guys. Um, we'll go up to Sand Hill Road and find out. I had to find out who the bio VCs were. I had no idea because there's virtually no crossover between tech and bio VCs.
Yeah.
And then I just found myself doing it, spending more and more time on it. And I got kind of fascinated in it. I'd always been fascinated back to engineering around DNA. When I got to Bell Labs, the Bell Lab story, the first thing I did was spend a year designing a computer inspired by DNA. I thought, hey, we do all this amazing computation happens inside DNA, and there are all sorts of equivalents of, like labels on packets and packet switches, and you can draw all these analogies. And I end up painstakingly designing a whole computer from the software all the way down to the hardware based on just long streams of characters. Like, just could you build it in DNA? So I'd
Yeah.
always been interested, got more and more interested in it. And then I remember having a meeting with a VC one day, and they said, we love this. We'd love to write a check, but we don't know who we're going to write the check to because the company doesn't have a CEO. And I thought, interesting. And went back and ended up handing over, like, spent about a year starting one company and doing a search for a replacement. And I think it was somewhere in '07, we started Helixis, which became that company, was real time PCR, as I said. And then within, I think, just under 3 years, we sold that to Illumina. And so in 2010, I ended up at Illumina, completely out of my depth. It was like a company that has just launched the HiSeq, sequencing was just taking off. They just bought the PCR company, and everyone was looking around going, why on earth did we just buy a PCR company when NGS is just taking off? That's another story in itself. And then had a very, very interesting time and ended up staying there 7 years. So that's one version of how I ended up in the life science side.
Yeah, actually, the that's how I know Alex. Well, we don't know each other well. I mean, I was never interactive with Alex outside of either this I guess outside of this podcast. But I mean, Alex overlapped a bit during my time at Illumina as well, and of course, Alex was like multiple times like on the hierarchy seniority level higher than I was. But I came in during roughly the time where Illumina started saying, "Hey, you know what? We we think we've got the chemistry down, but now we need to kind of help people with their data." So it was Alex who really shepherded in this product called BaseBase, which was a I guess the best way to describe it for folks that aren't familiar with it, it's like probably a software that can help analyze the data that comes off of the so-called DNA sequencers, which is sort of Illumina's bread and butter. So maybe to make it both general and a little bit deeper from a story perspective, Alex, like how did you think about creating these different organizations, almost like these strategic initiatives that you put forth at Illumina that were quite groundbreaking? I'm sure it was super hard. I don't even know how you justify ROI. I don't know what your how you justify the business case for any of these ideas of yours, because you shepherded both this base space idea, which is software, at a company that didn't sell software, and also this whole idea of population genetics, like sequencing not like a couple of people, but like hundreds of thousands of people back in a time where that just wasn't a thing that people thought was probably impossible, right? So how did you think about the building those type of initiatives? You know, how did you justify those?
You know, I'll separate those two, though, just to be clear. I'll take some credit for the first part. The population sequencing has been a long time dream, I would say, and so that's something I ended up taking over for the second half of my time at Illumina. But one way to think about base space is continuing, in terms of my biography. I turned up at Illumina. It was pretty clear, again, because it literally was in April 2010, and the HiSeq had been announced in January 2010. And the first thing was that BGI bought 128 units right out of the gate. So it was clear that sequencing was going to be it. And any thoughts of doing PCR at Illumina in early 2010 started not to make any sense. So I was like, well, I guess I'm not going to be doing this for very long. I better make myself useful. And the thing I could do, or I did have some understanding of, was computers. And I would look around the table at the executive staff meetings and go, boy, everyone here knows an incredible amount about biology and life science tools and chemistry. But there was less knowledge around what we might be able to do in terms of computation. And there were two things happening inside the company as I remember them. One was we knew we were going to build a very small sequencer, the MiSeq. And classically, we would use the mainframe versus personal computer metaphor. We've got mainframes, you put them in rooms, and there are people in white coats that take care of them. But then PCs were going to go, personal computers were going to go into everyone's house, and people had to be able to run them themselves. So the idea was when the MiSeq came out, we went, well, how will these institutions, it might be a professor at a small college, they don't have big armies of bioinformatics people. How will we provide bioinformatics for them? So we were thinking about that. And we were also thinking about the fact that we could see ahead to the one day doing something like the HiSeq X. So we knew that we would be able to have $1,000 genome. But back in 2010, if I remember the numbers correctly, we could see we could offer $1,000 genome, but then people would spend $1,000 doing the computational analysis of that genome. And the cloud seemed to fit the idea of perhaps solving both those things, being able to provide very kind of easy to set up, plug and play bioinformatics via the cloud, because you get both the software and the computation already set up for you. And then the second one was potentially, longer run, to be able to drive down the cost. And in the end, that's just Moore's law, right? The great thing about assembling a genome is it's a constant amount of computation. So Moore's law solves all problems there eventually. And you could kind of tell that Amazon clearly was going to lead to, was on a wonderful low cost trajectory for that computation. So it seemed like the best solution to both. And I would say my I think it was an original contribution at the time was coming into sequencing. It really felt like it was everyone used the phrase big data because it was in at the time, but it also was a huge big data problem, and everyone thought about having, very fast fiber optic networks feeding big disk arrays, and you'd have to put them in the same room as the big sequencer. And we just thought, it's just this big data problem. How would you get that up to the cloud, right? I mean, the cloud is, especially in those days, right? A fast internet link was, 100 megabits per second, let's say. And typically they're more in the, 10 megabits per second. And so it was this issue where the cloud doesn't feel like a good fit. You go, well, you got the cloud up there, but how are you going to get the data there? And I remember sitting in a staff a meeting and just listening because I didn't know anything. And I was thinking, well, yeah, the HiSeq does a whole human genome, which is around 100, let's call it 100 gigabytes. But it took at that point in time, I'm pretty sure, 2 weeks to do that genome, which seemed super fast at the time. But I just went, well, what's 100 gigabits divided by 2 weeks? And hope I have this math right. And it came out at something like 7 megabits per second. And I thought, oh, all we have to do is just stream the data up while it's coming off the machine. You can't do anything with it anyway till you've got it all in hand, right? And that's all we ended up doing. We just went, you know what, we'll just send these machines out into the world. And the first thing they do is they wake up and look for Illumina's cloud infrastructure. And if it finds it and it has permission from the user, it just starts streaming the data up there because it's just not that much data in the end. It turned out to be a pretty easy problem. So long story short, we ended up building base space to try and serve, it's always dangerous, both those needs. Originally for the kind of first time MiSeq user wanting more back-end bioinformatics, but also longer term, how are we going to make sure that we don't get caught in 2014 with $1,000 genome and $1,000 assembly and variant calling process.
Yes, before Costas jumps in and asks his big data, because you triggered him with the word big data. I'm sure he's going to have questions for that. But before he does that, I would it be possible for you to just educate the audience on the difference between, because you mentioned this word multiple times, PCR, and I sometimes I know we know what it is here, but maybe you could kind of elaborate on, let's say, PCR versus the technology that we're speaking about, this so-called next generation sequencing, and just maybe contrast those two for the audience first, and then I'm sure causes us the big data questions.
Well, that thanks for that, Les. That's one of my favorite questions because I've obviously worked in NGS, but also worked in PCR, and I think they're both amazing technologies. And but for a long time, and PCR is much older. I mean, the first versions of PCR was invented by Kerry Mullis in the 80s. Famously, got the Nobel Prize for it. Um, and and PCR is really a harnessing of nature. Going, DNA comes in a double helix, and cells are really, really good at replicating it. You kind of unzip it, you do some magic. Obviously, we won't go into it in detail here, but Mullis's genius was recognizing a way to do it in a test tube, that you could do it. You didn't need all the mechanisms of a cell to do PCR. And so he worked out that you could, with a very simple apparatus, you could magically replicate DNA. And that turns out to be so useful right across the world because in engineering terms, it's amplification. You just give me one of these things and I can multiply it by a billion because it doubles every time. And it's that classic, one piece of rice on the first square, two pieces of rice on the second chessboard square, and you end up like with a whole universe full of molecules by the time you get to the 64th square. And so PCR is just an amazing amplification effect of DNA inside a test tube, and you know the field has worked out just so many different ways to harness that. And I think most recently, most famously, it became famous during COVID because it's an amazing way to do a COVID test. And the COVID test is actually a PCR reaction which only fires up in a blood sample or a spit sample. It only fires up if it finds the very set sequence, like a subsequence of the viral genome of COVID-19. And then it just goes bam, and it makes a billion copies of it. And you can think of each one having a little tiny LED attached to it in the sense that a little bit of light suddenly turns into a massive amount of light. And that's the test. It's just replicating the DNA of the virus or RNA of the virus and then sufficient that there's enough light that comes out of the sample that it can be seen on a camera, and that's a positive test. NGS, on the other hand, is so I always say PCR is great when you know what you're looking for. If you've got a sequence, I have a DNA sequence, and it might be just 10 or 100 bases long, and I want to find out if it's in a sample. It's a quick and easy and incredibly sensitive and accurate way to do that. NGS is infinitely more complicated, in effect. Just one small step of NGS is a PCR step, for example, plus many, many other things. But it is able to actually determine the sequence. And I like to use this analogy. If it's the difference between reading a novel and using a search engine in a novel, right? NGS is reading the novel. Like, I haven't read it before. I'm going to read the whole thing. I'm going to learn lots. It's going to take me a lot of time. But if I want to know, does the word universe appear in the novel? How many times is it? I use a search engine, right? And that's what PCR is. So they're fundamentally different. They're very complementary. I think in particular in COVID, NGS decoded the COVID sequence, which enabled everyone to build a PCR test, which could then detect it all over the world. And if I don't know, a PCR test is at least 100, if not 1,000 times less expensive than an NGS equivalent test. But many would dispute that. But I would say let's say anywhere between 100 and 1,000 times cheaper.
Yeah, so you're right. I do have some big data questions, but I think it's the simplest one first was you were generating all of this new information. How receptive were the users to it? I mean, was it too much at the time? Was interpretation really yeah, even available at that time, or was there just so much being pushed on the recipients or the practitioners of that test to understand it, decipher it and to figure out how to apply it, whether it was too early to research or in the clinical setting. Because I think that's, I think that's where we are today. We have so much information, but how practical is it? And I guess if we take a step back and look at the early days, how did we convince people that they needed that information to really be on the leading edge?
Yeah, it's a super interesting question. I think first of all, the early customers were researchers, and they just love data, right? I always say this is just a thought experiment. I always say the first of every model of Illumina sequencer goes up to the Broad, and they don't want the manuals, they don't want Illumina software, they don't want any directions. They like just give it to us and get out of the way, right? And they're going to feed it into their vast computational farms and all their own software, and so they're just hungry for the data. And so I think researchers as the first customer, they're just, I would say, just in general in life science, they're wonderful first customers because your product can be completely prototypey and I don't want to use any four-letter words, just not very good. And people will love it because that's kind of what they do, right? And so that worked fine at the start. And there is this big arc, and I was fortunate enough to experience it while I was at Illumina from that point in 2010 when the NGS really kicked off. It was strictly speaking the second product, but really in effect the first product, and the clinical adoption. And that was huge, right? And so, and Illumina was very much a life science company, not a clinical company. And in those 7 years, there was so many challenges and so many false starts. And I remember when we started out, we thought, oh, it's easy. You just collect more records and send a big file to the FDA, and you're kind of you have a clinical product. We just didn't understand reimbursement. We didn't understand that you were serving patients and hospitals and doctors. That it was just such a completely different business than selling boxes to the Broad. And so that I feel like started the longest or biggest set of challenges around how do you go from huge variant files to a clinical report, which stereotypically will just be a PDF with things that a doctor, a physician would find useful. And I don't even know that there's a single answer to that other than many years. It continues, right? When I'm talking about things that were happening, well over a decade ago, as we went through that. And I think you see it now, right? I mean, doctors all the time are getting a Foundation One report, and they're starting to understand how to read it. And the reports, I think, have plugged in much more to the available therapies, and it's gradually coming clear. I don't think there's. I remember talking a couple of years ago, talking to a CSO at one of the very, very biggest biotech companies, and they were having trouble getting one of their companion diagnostics to the patients that needed it. And we were talking about them. We had all these theories. We were a startup talking to them, and they said, "Well, no, it's not. It's none of that." He said, "What happens is the patient comes in, and we'll run one of these tests, Guardant, Natera, Foundation on it. It comes back in as a PDF, and the PDFs get lost. And so they do the first line clinical, whatever's recommended first, right? But imagine the PDF from Guardant has lots of other suggestions. You should try this second, you should try this third. And that document is just lost because it didn't neatly plug into the EMR system, right? Where's it sitting? It's sitting at some other doctor, at the pathology lab or something like that. And that turned out to be the biggest problem. So it wasn't just creating the right document, it's making sure that the document somehow moves with the patient through the clinical healthcare system. And as we know, that's just an incredibly challenging and time-consuming thing that changes over a very long period of time.
Yeah, I think we've seen the same thing happen here in Canada. Some of the clinics that we work in, very siloed in terms of where that product gets spit out into and where it resides long term versus the clinicians who are using it on the front lines. There's a huge disconnect between those two. And I think the paper that you'd commented on recently about the UK's 100,000 genome project and the
Yes,
utility
fascinating.
of doing all those tests, and does it make a difference at the end of the day? I think one of the important things to pull from that was that you have to use the information to actual for it to matter, and if you don't, you're not really seeing a change in the clinical treatment and therefore potentially the clinical outcomes. um, I don't know if you want to touch on that a little bit, because I think that's a huge experiment that is still ongoing, and I think there may be some skeptics that are starting to arise based on the output from that study.
Well, that thank you for bringing that up, Costas. That ties into something Lester asked earlier, which is the second half of my time at Illumina. I was responsible for population sequencing. And so that was a very different, I would call that almost business development role for me. If the first one was sort of a technical leadership, this was very, very different. It was absolutely fascinating. I was thinking about it just yesterday. I got to have dinner with the Australian Prime Minister in his house, which was such a thrill for me, because people were fascinated by these large scale programs. And the
Mhm.
1,000 genome program in the UK was the very first one. Celexa, which Illumina bought for the sequencing technology, was out of the UK, or elements of it were out of the UK. And so Illumina's always had very deep roots in the UK. David Cameron, who I believe was the prime minister in that period of time, lost a child to a genetically inherited disease. So he was very interested in sequencing and what could be done. So a lot of things came together in that 100K genome program, and it was the first of its kind, and in many ways, way ahead of its time. And I remember how excited we were about it at the time, but it was based on something even an engineer could understand, which was the idea that a person has cancer and there's a tumor. Let's take a sample of the tumor and look at the DNA sequence of the tumor. And then let's take a sample of their healthy cells, could just be a blood cell, look at that DNA and just compare the two. And it's very much an engineering or software thing, right? Like I'm going to get two strings of bits and I'm just going to compare them. And most of the time they'll be exactly the same. But occasionally one of them will be different. And you'll immediately go, that's a variant that's somehow involved with the cancer. And
Mhm.
so it seemed like a very elegant way to work out what was happening with cancers. And I guess it was, but it turned out to be, not turned out to be, it was a very expensive way to do things because back then, first of all, the DNA sequence was inherently expensive. I'm going to round it to $1,000, right? But it was probably even more than that. Could have even been as much as 5,000. But it turned out that to get um, the right level of resolution of the genome, because there's a lot of chaos in the cancer cells, you kind of had to do three whole runs on the cancer genome and one on the healthy genome. And so you were doing four whole human genomes for every patient. And then you were taking all that data. And again, this was a long time ago, so computation was expensive. And you were going to have to do those comparisons. And all you're going to get out of that is some huge jumble of variants, some of which are noise, some of which were in the non-coding regions, which no one knew what to do with. In fact, most of them probably were. I don't remember. And so working out which of those variants was just an act of discovery. And in that sense, it was incredibly important in terms of the science that was being done. And that paper you just referred to, the interesting element to it, though, was that project involved the NHS. And the whole idea was we'll find, I think it was 50-50 between rare disease and cancer patients. So, order of 50,000 cancer patients. And we will try and help them with this process, which of course you want to do, right? Everyone wanted to do. And it probably helped with the justification for the funding. It was, I think, in the end, around a $400 million program over there. But in that paper, it turned out that once you put aside a lot of different things and get to the core of the matter, only about one in every 200 of those patients was actually got a change to the treatment or the therapy that they were originally getting, because they were all in treatment. Otherwise, they wouldn't have been put in the program. The cancer had been detected. So it turned out that the influence was small. But again, this is so long ago. It was tumor normal, which is now not done to the best of my knowledge. Maybe it is, but we don't hear about it anymore. And so the whole field has moved on. So I'm not quite sure what to make of that result. It's distressing in some ways, but thought of in context, it's not clear to me what the lesson, frankly, is in the current time, just because so much has changed, whether it be the sequencing itself, the computation, the science, the economics, the available therapies, the healthcare system. Everything's moved along basically over a 5 to 10 year period from when the last of those sequences was actually taken.
Yeah, it's fascinating to hear this historical context that you put these different initiatives that I was just tangentially a part of. So it's really great to hear kind of the history and the background behind them, and just hearing you kind of speak about a lot of the. It seems like at least from the at least Illumina side of things, the technology, the brilliance of the technology really was a thing that pulled in like early adopters, like abroad, like others, right? Even say like the UK from a whole genome population sequencing perspective, even drew in like the Australian Prime Minister, who was very interested as well, right? Like just many people were interested based on just the technology itself. So now that maybe some of that has, I wouldn't say dwindled, but maybe that has created results that maybe weren't as simple as people would have thought it would be, like the paper that Costas kind of referenced. How do you, as an advisor to other genomic organizations, genomic companies or other biotech companies, how do you advise them on like how should if someone wanted to start a company today, or they have a company and want to deliver value to their customers, like how should they think about this particular space and how they should deliver value? Because I don't know if you is it just purely based on, and I don't believe this to be the case, so maybe I'm leading the witness here a little bit, but it can't just be, oh, we'll just create like cheaper, like we'll just do the sequencing constantly cheaper and cheaper and cheaper, and that's the answer, because it doesn't seem like what you're saying doesn't seem like that's the problem, at least that you see. So maybe if you could kind of walk someone through, if I was just a new entrepreneur in this space, want to start a company, or someone who's in this space already has a company, how do you advise me to, hey, move forward in this changing landscape?
Oh my gosh, I can talk for 9 hours on that, but I promise I won't.
Perfect.
Another
Perfect.
great. Um, just thinking it through. I want to use two examples that are almost completely opposite. So I think it shows the richness of the opportunity. So one in particular is a company I'm on the board of called GenCove, which is in New York City, and some folks out of the New York Genome Center that were very interested in this. Right? Remember, I just talked about how much sequencing was done for cancer tumor normals. The funny thing embedded in that, for those not familiar, is a normal. Let's call it a normal genome sequence. The sequencer runs through the same area 30 times, and because it's a fairly error-prone process, and so it's, if you will, you're going to do it 30 times and then look at that particular location along the genome and just check, did I get a lot of C's? Did I get a lot of G's? What happened there? And then we'll call the base and say, no, that's a G. And so um, these folks recognize that there's a lot of redundancy in that process. And so there's this thing called low pass sequencing that says, why do we do it 30 times all the time? Let's just do it once. And the amazing thing about most genomes, when you think about it, is every time you get a new genome, it kind of looks a lot like the old genomes, right? If you're getting I don't know, if you're getting a cat genome and you sequence another cat, your own cat, it's, I don't know what the numbers are. It's something like 99% exactly the same as every other cat. It's probably more than that. So it's only very small changes. And so, long story short, with GenCove, they recognized that there was a huge market in agriculture for sequencing. But agriculture is a very cost sensitive business, and sequencing to 30x was just too expensive. So they built the whole technology and the offering, which is primarily cloud based, around the idea of just do a 1x sequence, which is very inexpensive, and we'll take the relatively noisy genome and we'll use informatics to clean it up. And yes, you may not want to prescribe a cancer diagnostic on it, but if I'm doing a plan for cattle breeding, it's perfectly great. And I would draw that distinction. It has applications in human, I would say less diagnostic, but for example, in human, large-scale human programs where you're looking at huge amounts of data and processing that data because you have such a broad averaging and denoising effect in the data. And that company is doing really well and is being amazingly effective in ag. So it's super fun to go to the board meetings because you're often talking about cattle breeding and all the weird elements of that. It used to be that they would only genotype the bull because I should get this number, but you have one bull for every, I don't know, 1,000 cows or something like that. So you kind of get half the genome that way. But at a certain price point, they want to do all the cows as well as the bull. And we're crossing over into that point now. So it's a fabulously, from a business point of view, fabulously inelastic. It's also driven by the cost of genome sequencing, which, as you know, has been radically dropping in the last couple of years as competition comes in for Illumina. And that's helped that business as well. You get to ride those curves. All the work and all the competition that happens accrues to you as a provider of sequencing on that side. The other extreme, in a way, from that is proteomics. I'm fascinated by that, probably as a new convert, again, as an engineer discovering these things at the same time as all the other non-experts. That the central dogma of biology, I guess, is that you've got DNA, and the DNA gets processed into RNA, which gets processed into proteins, and everything entire body is built up of proteins. And for all sorts of interesting reasons, and the sequences of amino acids, and for all sorts of reasons, it's incredibly difficult to sequence them. And we thought DNA sequencing was the miracle, but now DNA sequencing has become every day, so that's a hard business to compete in. As we see, you've got obviously Illumina as a very unusually strong monopoly, and some very well-run companies like Element are proving that you can compete against Illumina, but so many have fallen by the wayside and never made a profit. And it's just a very, very difficult business. But proteomics is now an open field. And the expectation is that potentially is a much bigger market than genomics, right? That now you go, no, I'm not just analyzing the plan for the building, but I'm analyzing the whole building itself, the materials in the building, what are the walls made out of, how much moisture is in the concrete. It's sort of an infinite analogy. And so I'm on the board of am I board? No, I'm an advisor to a couple of companies that have very innovative ways of doing proteome sequencing. And so I'm very excited to see how that comes out. And I think you're seeing more and more activity. We saw Thermo Fisher buy Olink within the last 12 months for $3.4 billion. And so you're starting to see really strong interest in it. That might be an impact of if the antitrust environment does change with this new administration. Wouldn't surprise me if Illumina went and bought a proteomics company, for example.
Yeah, no, wonderful. Yeah, thanks. Just for myself, just to kind of not summarize, but take your those two examples. So I guess as an entrepreneur, if one's an entrepreneur and they're starting a company or they're an entrepreneur with a company, things that they should do is find a problem that that's useful when you solve it, it's useful, like the agriculture, a unique problem, right, that can be solved. Or from like the proteomics space that you kind of talk about, it's like finding a big tailwind. It's like you suggested, riding a wave. If you know the wave coming, you might as well catch the wave and not like go where there is no wave, right? And try to create a wave would be kind of a little bit more challenging to do. So I appreciate those kind of like I appreciate that advice.
That's very well put. I just say the way I try to think about these things is where does the money come from for your revenue? And the great Genco's a great example of that because it's a pre-existing business which is completely served right now by a completely different technology called microarrays, which was the business that Illumina was originally built around. And so the agricultural industry and consumer business like 23andMe and Ancestry was all based on Illumina microarrays, and though not widely advertised as such. And what Gencov is doing is providing a better data set at a lower price. And so the great thing is you know exactly where the money's come from. you've got to go find someone that's buying microarrays right now and get them to stop buying microarrays and buy GenCove low pass sequencing instead. And that's kind of the I'm going to say the easy business model, but at least you know exactly what your target is, and so it's very useful. And the money's already flowing, right? People have budgets every year, and it's about a depending on who you ask, a $2 billion or a $4 billion business. So it's a lot of money flowing through, and it's dominated by Illumina first, Thermo Fisher second, and Agilent third. And those three are probably the vast majority of that $2 billion. If you go to build a proteomics company, that's completely different because I don't know where that money's coming from because no one's spending any money yet to speak of on proteomics, right? And those back to the early adoption of genomics, it starts off on research, right? So it starts with um, principal investigators writing grant applications and getting them funded. And then they're going, well, I want the best, I want a paper in Nature. So I want whoever's got the best proteomic sequencer, I want it. I don't care if it has sides or if it leaks or if there's an instruction manual back to that thing. I just want the best one. And that gets that next cycle going. You know, if you go, it comes in cycles. First of all, we had microarrays. Actually, first of all, we had PCR, then we had microarrays, then we had NGS. And I think there's a growing consensus that the next thing is proteomic sequencing. But that engine takes a long time because you've got to spin up the money engine with people that are spending money on proteomics, and that's just not material yet.
You know, the one thing I didn't hear in there was AI, the application of AI to this space. Is that just too far off, or is it just it's just not really necessary in this space at this point in time? Where do you land on AI and diagnostics?
Well, I'm very biased. I did my PhD in AI back in the 1980s. So, a very, very retro version of AI. It was called Expert Systems, and
Hmm.
um, it was super fun. I only know one programming language, which is Lisp, that I think has pretty much vanished from the face of the earth. Um, so I've always been interested in AI, and a friend and I a year ago started a company called Ryght, RYGHT. I spent a while a few years ago on the board of a clinical research organization, a sort of I think it was a 20,000 person clinical trial runner, and so got exposure to clinical trials. And I felt as the large language models came online, they seemed a natural to build tools for the clinical trial business because it's an incredibly people intensive, expensive, and slow
Hmm.
business. And everything is challenging. In particular, identifying patients is challenging, but also keeping them engaged is challenging. And so that's been a really interesting ride. So it's a little out of the category. So it's healthcare, but it's certainly not directly tied to genome sequencing. Um, the area that I'm most fascinated about intellectually, where it is having an impact there, is the fact that it seems the most extraordinary thing to me that these large learning models, even though we think of them in general as they're called language models, but we think of it as human
Yeah.
language, that they're magically capable for reasons it seems we don't completely understand of reasoning with human language. They're really good at dealing with the languages of the proteome and the genome, and that you can
Hmm.
train them on these things and they will start to give you new ones. For example, there's basically an infinite number of proteins, as I understand it. The number of
Yeah.
variations on protein sequences is effectively infinite. And then there's folding and there's post-translational modifications. You know, it's a vast space. And so the abilities of these instruments to, sorry, these models to generate new proteins, which in turn um, potentially drugs, is absolutely unbounded. And linking that back to instruments to me is interesting, and I'm really working hard on understanding this thesis. But why I think it's another amazing potential driver for the tools business is we know in human space that those models are incredibly difficult to train. You know, a baby gets trained in, just a few years, just crawling around the house, right? But we seem to have, for these foundational models, we have to seem to take all of human knowledge and pump it into the model, like a giant fire hose, right? They're just, for whatever reason,
Yeah.
Jan LeCun makes a big deal out of this. For whatever reason, they're kind of really stupid when it comes to learning. and different, suggests that they're actually learning in a very different way to the fact that humans do. This is also true of these genomics and proteomics models. So where, I mean, it turned out to be pretty convenient that we had the internet and we had all this data and Wikipedia and everything else was already out there when we needed to start training large language models. We don't have that for genomics and proteomics. And so I think
Yeah.
it's a huge opportunity for building the training data sets. And that can be as rich as plugging it into EMR. So you have the outcomes, right? LLMs eventually, they speak English, sorry, any language, French, really well. And so they can start to go through doctors' notes. They can recognize handwritings, right? So all of a sudden you both unlocking a lot of the phenotypical data that's in the EMRs with some flavors of LLMs, but also being able to link that to vast bodies of information that you need to be able to do training. And then on the back end, as the LLMs start to suggest new drug candidates, you've got to test them. I joined the board recently of a UK company called Sphere Fluidics. And Sphere encapsulates individual cells into tiny little bubbles and can run basically 40 million experiments a day on individual cells. And that's interesting when you're starting to get the output of some of these AI drug development companies. How do you test en masse, right? I was fascinated when I first got into life science to realize that most work was still done on 96 well plates with pipettes. And automation means a robot to do the pipetting. It's fascinating to me that and now I understand better the reasons for it, but it feels weirdly baroque. And I think, and there's been a lot of experimentation with new models for how to do, much greater volumes. And sometimes they work and sometimes they don't. But I think those two things, I think developing tools to develop training sets and tools to do validation of candidates to match the likely incredible hunger and output of these large learning models, I think is a giant potential opportunity that's building up right now.
And you truly are honorary Canadian. You recognize that we are a bilingual country, French and English. So, unfortunately, neither of me or Costas are very fluent in French, but on Paul, un petit peu le français, just a petit peu.
Very nice.
Yeah,
But
I won't even
you
try
know,
my terrible French.
yeah, actually, the thing that came to me is, as I hear AIs, and they're the versatility of really using any language. I almost feel like you need to go back and re-examine your DNA computer. I mean, realistically, they can actually now sort of maybe meet the sort of the requirements of speaking in a DNA based language. I don't know how many base
I
pairs
think
that's
that's
going to
it.
be.
I was drawn to it because, and still am. It's kind of weird, but there's quite a bit of activity still around using DNA for storage, which I drives me crazy because I don't understand it, and I strongly disagree with it. I feel like DNA is like the worst system for doing storage, but that's again the whole 9 hours of talk. But doing computation, it's great because
Yeah.
computation is all about parallelization, right? And so and we've gotten so good, as I mentioned earlier, at synthesizing DNA into specific sequences that you can use for sorting it, labeling it, and sorting it, and replicating it. And so
Yeah.
the interesting thing about PCR is if you can imagine if you can encode a computation in DNA, you can PCR it and you can make it'll double every hour or something like that, or every 3 hours, right? And so you get this constant. Sorry, that's wrong. But anyway, you get massive replication, and so you can get parallelism, and it's also incredibly energy efficient, right? PCR uses effectively no energy, and so it's doing bazillion transactions a second and using no energy. And so if you can work out how to harness that, it's a very, very powerful computational engine, I suspect.
Well, I think that reducing computational time and with lower power, we can now label that Dickinson's law instead of Moore's law. So, if anyone is listening out there and they're
For our
looking
billion
for,
listeners, yes, thank you.
there you go. We've coined a new law here as well.
yes, and actually, we may have, I know we already took a lot of your time, and you've been very generous with your time and obviously all your experience and stories. But maybe maybe we will end off with the your time at Bell Labs. We spoke a lot about research and the DNA computer that you were at least conducting experiments on, and maybe you could speak to people, let people understand like why was Bell Labs so special. like what made it special, at least during the time you were there? And do you still have fond? I assume you still have fond memories of your time at Bell Labs.
Yeah, it was great. I was first there as a student. I was doing my PhD, and I spent a year working there. And I got there, and it reminded me of my PhD in that they pretty much gave you a room and said, kind of go for it. There was no direction or we want you working on this project. And I noticed opposite my little office in that giant, completely windowless building was another little office that had a very strange looking, unusual looking computer in it. And I asked about it, and they said, "Oh, that's a Lisp machine." And I didn't know anything about Lisp or AI or anything, but someone there just bought, and these were customized machines that were built. The whole CPU architecture and everything was built in to just run the Lisp language. And they just had a Lisp machine there. So I just went in and started playing with the Lisp machine and taught myself Lisp and worked things out and started designing VLSI design tools using Lisp. And because this was a VLSI design, chip design department. So it's what, and that was my background. That was my PhD. And that in the end led to what I did in the PhD because they happened to have those machines just laying around and no one using them and everyone just encouraging them. It's like, oh yeah, just go play with it. Spend as much time as you want on it. I went back later for a full-time job after I finished my PhD in Australia and worked for an amazing guy called Alan Wong, who had this vision for building an optical computer. And it's a little, I think I channeled him a little bit when I was giving you guys the biological, the DNA computer pitch. Alan was like, it's ridiculous that computers run on electrons. It's like it's the worst way for a computer to run. And he would say things like, think of a computer, electron computer is like New York City. You know, all the cars have to stop at the intersections because they all interfere. But if it was an optical computer, the cars would just pass through each other at all the intersections, and everyone would go, "Whoa!" And he was a very compelling, and he made all these arguments, and he convinced me. So I went back there to build an optical computer. I'm like, "Oh, electronic computers, that's ridiculous. I can't believe we're so backwards." So in parallel with working on that, just a vision or an architecture for a biological computer, that was just in a way a side project for me. The project we were all drawn to was to build an optical computer, which turned out not to be a great idea, again for reasons I won't get into. But then I bounced back into chips and was really interested in the idea of the fact that chips were light sensitive. And back then, this is now the early 90s, that image sensors were all built in a process called, oh my God, I'm getting so old now, I can't remember the name of it. How funny. CCDs, charge coupled devices. And so your camcorder had a CCD in it, and it did a great job for that. But we were thinking, well, we had a lot of access to CMOS, like standard chip fabrication. Maybe we could build cameras that way. And Eric Fossum, who was at JPL, was really at the center of that activity, but also the University of Edinburgh. And so a lot of us started to work on the idea of building image sensors out of CMOS chips, which turned in, we got a lot of patents and turned into like every image sensor, ones we're using right now, they're all CMOS, right? It just in the end made perfect sense to integrate the electronics right alongside with the photosensors. CCDs couldn't do that. You couldn't build a transistor in a CCD process. And then that in turn, there was some security people at Bell Labs, because Bell Labs and AT&T were in everything, that were thinking of, was there a better way to build a fingerprint sensor? And I ended up talking to them, and they were like, "Well, you're building these really compact optical sensors. Maybe we can build use that to build a better fingerprint sensor because you're looking at a fingerprint." And somewhere along the way, we got the idea of, well, why don't we just get rid of the optical part and just put the finger right down on top of the chip and use the circuits inside the chip to measure the capacitance? Because if you're a chip looking up at a finger, sometimes you've got skin coming right down to the surface, and sometimes you've got a big valley as the skin on the valley in the fingerprint reaches away. And it turned out, I'll never forget it. We went and built that chip, and it went to the fab. It takes months. It comes back. We put it into the big test system, and I put my finger on it, and an image came out. I couldn't believe it. It worked like first time. It was just amazing. And that we filed the patent for that, I think, in '95, and that eventually ended up in Apple's hands and as Touch ID, which ended up in the phones for
Wow.
the first time. So I just give you all those just to give you some idea of the variation. We just wandered around and we did what we thought was kind of interesting and published papers. And we were measured not by our, I don't know, call it useful commercial output. We were just measured by, did you get some good publications in the last quarter? And are people being attracted to your work, right? That was a good thing. If you became a sort of a catalyst for effort, that was a really good sign.
Mm.
And right beneath me, far more historic, right beneath me, there was a group working on neural networks, and one of the lead people there was Jan LeCun, who's ended up heading up AI for Facebook, Meta, and being very famous in the field. Um, and we were like the chip guys. And we thought they were kind of crazy down there. It's like because we couldn't do any math. We didn't have the vision, so we couldn't do any math where we thought that would ever be useful. It's like, yeah, it's cute. Like he's built like 3 neurons. Like fun. So completely missed that one. I got to say.
Wow. Wow. Actually, you know what, Alex, I lied. I'm going to ask one more question if you is so generous with your time here. One thing that we, the publications, or by publication, you mean like newspapers, have said about Canada over the last maybe year or so, maybe a little bit more, is that Canada has a productivity problem, right? And they usually measure by like GDP per capita, right? So the Canadian government has put in a lot of money, maybe not as much as Meta nor Google into AI infrastructure, right? About $2 billion, which I guess compared to Meta, that's like a Wednesday or something like that, right? So it's so how would you, and you being a part of these various pop gen initiatives and also developing such foundational and underlying technologies at Bell Labs, like how would you advise like a government such as like the Canadian government, how they should think about investing to for the future of say for Canadians in Canada in terms of innovation? Because right now, for instance, one of our larger population sequencing type of initiatives is 15,000 genomes, right? Which is like it compared to like what you shepherded and your team has shepherded at UK is just significantly smaller, right? So what would you advise? How should the government think about investing into the future of innovation in Canada?
Well, with the caveat that I feel appreciate the question, but I feel hugely ill-equipped to answer it. I think maybe one lens I could view it through, which I'm more familiar with, is Australia, right, which has similar scale challenges.
Mhm.
You have the additional challenge of being right next door to the US, for all the good nil that brings. Um, but I think it feels a little obvious and superficial, but it's the only answer I really know of, is finding out what the nation's unique differentiator is. And certainly, I remember in Australia, a lot of the conversations we had there was about the advantages of being a single payer healthcare system with an integrated EMR system. It's kind of obvious that that's a challenge in the US, right? And we were alluding to this earlier around, getting things into EMRs and making changes and everything like that. I think in the UK case, probably that was part of the 100,000 genome program. Justification was the sense that here's a single payer, pretty much integrated health system. We should be able to run a really good program like that. And that also showed up around COVID, right? The UK built a great COVID and continues to build a great COVID surveillance system. The US is not well configured for those kind of top down centralized projects. Um, and I saw a lot of that when I was doing population sequencing. I spent a weird amount of time in Beijing seeing how the Chinese government was working on population sequencing, and then going to Washington, D.C., and helping get the what became the All of Us program running at the NIH, right? And seeing the difference in the decision making process and everything else. China was able to act very fast. It felt like the U.S. decision making process. And we say you walk under those tunnels underneath the office buildings in Congress, and it feels like these decisions are made of millions of conversations, just constant conversations between visitors, aides, lobbyists, Congress people, the backwards and forwards with the administration to the Senate. China, it's kind of simple. They would go, oh, we do a 5-year plan, and we're going to make genomics one of the big 5 items, and go find the 5 most important professors and tell them to design a program, give us a budget, and we'll start pumping money into it. It's kind of a very, very different way of doing it. And I'm not judging them as a better or worse. They clearly both have different advantages and disadvantages. But I'm looping a bit. But I think for Canada, it's a matter of working out. It's like a business, really. Like, what's my differentiation? Because otherwise, the other place next door is going to take it over, right? They have to. U.S. is always going to have the scale and always have the money. Money in terms of people, intellectual property, and money. So working out a path in any of these areas just requires working very hard on differentiation, in my opinion, and not trying to do a me too. You don't do the 100,000 genome program for Canada. You've got to find out something that's different if you possibly can.
Great. Thanks a lot, Alex. That's really, really great. I think words of advice. It doesn't mean you're just spoon feeding the answer. We have to do the hard work. Is at least what I hear from that, and I appreciate that very much, Alex.
I don't know. Any other, I think is the question.
Any questions? Any questions? Any questions for you, Costas, or wrap things up? I know we took a lot of Alex's time already.
Yeah, no, I appreciate the insights and the little bit of the history, because I think people don't necessarily appreciate what they have today and what it took to build, and sort of the incremental sort of innovation that has occurred to get us to where we are today.
Great. Thanks a lot, Alex. So if you want to follow Alex to see more of his work, so you can follow Alex on X, or formerly known as Twitter, at Alex G. Dickinson. That's Alex G, and then Dickinson, spelled Dickinson. And of course, you are again the amazing LinkedIn provocateur. You can follow Alex in LinkedIn as well, and it's Alex Dickinson, and you'll be able to follow a lot of his, at least, posts and a lot of his thoughts along the genomics industry. And of course, if you are interested in connecting with us and also have any feedback on this podcast or any questions, feel free to subscribe, connect with us, and also comment below. Any other place people can find your work, Alex? I was trying to find a website or something that may have some of your work as well. Is Twitter and LinkedIn sort of best ways to follow your work?
Yeah, and I would even say it's narrow in that I rarely post on Twitter. I really, for whatever reason, I don't know why, I just am comfortable on LinkedIn, and feels a little boomerish, but I enjoy it. I like it because it's a very specific audience, and so I feel like I was really pleased to talk to a friend recently who said, Alex, I don't really read your posts. I just go to the comments. And I thought, he wants to see the discussion, and I'm always thrilled when someone, a CEO or a sequencing CEO or someone pops their head in, and you end up having really interesting insights and information. They're usually correcting me, but whatever it takes.
Great. Thanks a lot, and everyone else, have a wonderful day.
Thanks, guys. Thanks, Costas. Thanks, Lester. A lot of fun.
Bye, everyone.